Hi, I'm Dr. Sierra Cower, and I work at the Center for Neuroscience and Regeneration Research as a health science specialist for the U.S. Department of Veterans Affairs and an instructor for Yale University. My lab's primary research goal is to understand the mechanisms that underlie spinal cord injury-induced hyperexcitability disorders, such as pain and spasticity. Our previous research has found that the hyperexcitability and spasticity is in part due to abnormal dendritic spine morphology on alpha motor neurons. Specifically, we found a conservation of structural motifs in dendritic spines that closely accompany spasticity. These are spine density, spatial distribution, and form. We've also found a convergence of the role of the RAC1-PAC1 signaling pathway for these morphological and functional aspects within spasticity. Over the last decade or so, the preponderance of evidence is that hyperexcitability disorder is maintained, stored, hardwired by abnormal dendritic spine remodeling after SCI, and that dendritic spine structure predicts therapeutic efficacy. Therefore, in our recently published article in the Journal of Physiology, we explored the use of a PAC1 inhibitor, Romidepsin, for the treatment of SCI-induced spasticity. In this study, we performed a 50 kilodyne contusion at thoracic level 11 in mice expressing YFP under a Thi1 promoter. Mice then received 3 days of IP Romidepsin or vehicle treatment. We used an EMG paired pulse H-reflex testing paradigm and found that SCI resulted in a loss of H-reflex rate-dependent depression. For within subjects, we looked at the H-reflex at baseline, before treatment, and after treatment, and we found that Romidepsin treatment restored rate-dependent depression for several of the interpulse intervals. We then examined differences between sham and SCI subjects that were tested after treatment on day post-injury 29. We found that Romidepsin treatment in SCI subjects reduced the H-reflex compared to vehicle-treated subjects to sham levels for several of the interpulse intervals. Finally, we analyzed YFP-positive motor neurons from the lumbar spinal cord. We found that SCI increased the abnormal dendritic spine profile in vehicle-treated subjects, but Romidepsin treatment resulted in a reduction of total spines, thin spines, and mushroom-shaped spines. To summarize, Romidepsin, a repurposed FDA-approved PAC1 inhibitor, corrects abnormal spine morphology on alpha motor neurons and reduces hyperreflexia. Therefore, we will be continuing our research into the therapeutic efficacy of Romidepsin against SCI-induced spasticity. Thank you for taking the time to learn more about our study published in the Journal of Physiology. The full paper was published in Volume 602, Issue 19. Please contact Dr. Sierra Cower at sierra.cower@yale.edu, respectively, if you have any questions. Thank you.